The topic taken today for discussion is cell theory and prokaryotic cells. Cell is the basic unit of life. All the living organisms are made of either one cell or more than one cell. That is, organisms are either unicellular or acellular and multicellular. The term cell was coined by Robert Hooke in his book known as Micrographia, published in the year 1665. Then, a living cell was for the first time described by Anton von Leeuwenhoek, and that cell is a living verticella, a protozoan, in the year 1702. Then, the cell theory was coined by or proposed by two scientists, namely Matthias Schleiden, a German botanist, and Theodor Schwann, a British zoologist. These two people are credited with the cell theory. Schleiden, in the year 1838, found that all the plants are made of cells. In the next year, that is 1839, Schwann also proposed the same theory for animals. He studied different types of animals and found that all the animals are made of only cells. In this way, the credit for proposing cell theory goes to Schleiden and Schwann. Schwann also made two more observations. He observed that the plant cells are covered by a cell wall, and all the cells of both plants and animals are covered by a plasma membrane or a cell membrane. The cell wall lies outside the plasma membrane. Then, how the new cells arise from existing cells was not proposed by them. It was left to a German biologist known as Rudolf Virchow. He proposed the famous statement, omnis cellula a cellula. It means new cells always arise from pre-existing cells. So, for the first time, Virchow explained that cells do not arise de novo. They do not arise from non-living substances. Cells always come from pre-existing cells. Then, there are three tenets in cell theory. They are: all living organisms are composed of one or more cells. Cell is the most basic unit of life, and all cells arise only from living cells. These are the three tenets of cell theory. Then, cells are two types, depending upon the presence or absence of a nuclear membrane. If a nuclear membrane is present, the cells are known as eukaryotic. If a nuclear membrane is absent, the cells are known as prokaryotic. Prokaryotic means before the nucleus. Eukaryotic means true nucleus. All the organisms belonging to the kingdom Monera are prokaryotes. They do not have a nuclear membrane, and their chromosome is naked. Naked in the sense, the chromosome of bacteria lacks proteins, RNA, and also a nuclear envelope. In the case of eukaryotes, the DNA is associated with RNA, histone proteins, non-histone proteins, and surrounded by a nuclear membrane. Then, so in this way, organisms are either prokaryotic and eukaryotic. Bacteria are prokaryotes, whereas the organisms belonging to kingdoms Protista, Animalia, Plantae, and fungi are all eukaryotes. They have a nuclear membrane around their chromosomes. In this way, all the organisms are broadly divided into prokaryotes and eukaryotes. Then, in all the cases of prokaryotes and eukaryotes, or the cells of all prokaryotes and eukaryotes are surrounded by a cell membrane known as plasma lemma or plasma membrane. The structure of the plasma membrane is similar in all the prokaryotes and eukaryotes. I will be giving a detailed structure of the plasma membrane in the next topic. Then, the next point is, in the prokaryotes, membrane-bound cell organelles are absent. There are membrane-bound cell organelles like chloroplasts, mitochondria, lysosomes, Golgi bodies, like that. These are absent in prokaryotes but well developed in eukaryotes. Then, there are ribosomes known as 70S type. Actually, ribosomes are two types, 70S type and 80S type. Here, S means Swedberg unit. 70S ribosomes are present in both prokaryotes and eukaryotes. In the case of prokaryotes, the ribosomes are scattered in the cytoplasm of the cell. In eukaryotes, the 70S ribosomes are confined to mitochondria and chloroplasts. There is a theory stating that the chloroplasts and mitochondria were once free-living bacteria. By chance, they entered the eukaryotic cells as parasites. Later on, they became symbionts, and ultimately, they ended up as cell organelles. This is the reason to explain the presence of 70S ribosomes in only mitochondria and chloroplasts. Whereas in all the eukaryotes, in the cytoplasm, we come across ribosomes of 80S type. The 80S type ribosomes are either free in the cytoplasm or membrane bound. They are attached to the nuclear membrane, endoplasmic reticulum, and they are also, I have told you, scattered in cytoplasm. Sometimes the ribosomes are found in groups participating in protein synthesis. Then they are known as polysomes or polyribosomes. Both 70S and 80S ribosomes are made of two units. The 70S ribosomes are made of two subunits. They are 50S and 30S. See, the combined value of these subunits is more than the value of the complete ribosome. The 80S ribosomes are made of two subunits with 60S and 40S subunits. In this way, ribosomes are present in both prokaryotes and eukaryotes, and their main function is to participate in protein synthesis. Then, coming to the cell size. The size of the cells varies greatly. The smallest cells are those of mycoplasma. Mycoplasma belong to Munira. Mycoplasma are parasites. They lack a cell wall and they measure only 0.3 microns. Bacteria are also very small, but they are bigger than mycoplasmas. They have a cell wall and they measure 3 to 5 microns. Whereas our RBC, red blood corpuscles, they have a diameter of 7 microns. Then, the largest cell is the egg of ostrich. The egg of ostrich is a single cell and it measures about 13 centimeters long. Then, the longest cells are our nerve cells. Longest nerve cells are found in the neck of giraffe. In this way, the cells are of various types. Then, coming to the prokaryotic cell, I have drawn one prokaryotic cell here. I have told you prokaryotes have no nuclear membrane. Their DNA or chromosome is naked. The Prokaryotes are bacteria, blue-green algae, mycoplasmas, PPLOs. PPLO means pluro-pneumonia-like organisms. These mycoplasmas and PPLOs are parasites in human beings and other animals. Then, based upon the shape of the cell, the bacteria are classified into 4 categories. These 4 categories are bacilli, these are rod-shaped bacteria. Cocci, these are spherical bacteria. Vibrios, comma-shaped bacteria. Spirali, spiral-shaped bacteria. In this way, based upon their shape, bacteria are 4 types: bacilli, cocci, vibrios, and spirali. Then, prokaryotes. The bacteria also have a cell wall, but the cell wall of bacteria is different from the cell wall of fungi and plants. In plants, cell wall is made of cellulose. In fungi, it is made of chitin. But here, it is made of a substance known as peptidoglycan or murine. Peptidoglycan or murine is different from cellulose. Then, the bacteria are classified into two categories based upon the strain they take up. A scientist called Graham developed a stain to color the bacteria. Some bacteria take up the stain, some bacteria do not take up the stain. The bacteria that take up the stain are known as gram positive. The bacteria that do not take stain are known as gram negative bacteria. In this way, bacteria are classified into gram positive and gram negative. Then, the bacteria are covered by a cell envelope. The cell envelope consists of 3 layers. Outermost, the glycocalyx, this one. The glycocalyx is a loose structure, or sometimes it is hardened into a capsule. Capsule provides protection to the bacterium from the antibiotics. The middle layer is known as cell wall. I have told you already, it is made of peptidoglycan or murine. The innermost layer is plasma membrane or cell membrane. This is cell membrane. The unique feature of bacteria is that the cell membrane produces a number of inward foldings like this. As a result, the cell membrane is greatly extended. These infoldings are in various shapes. They are in the form of vesicles, tubules, saccules, etc. The inward foldings of cell membrane are known as mesosomes. Very, very important point, mesosomes. The mesosomes participate in various functions. They help in cell division. They help in the formation of cell wall. They help in respiration. They help in nitrogen fixation. They help in photosynthesis, etc. So they carry out a variety of functions in bacteria. So I have drawn here one structure, this is photosynthetic membrane. In the case of autotrophic bacteria, blue-green algae or cyanobacteria, the mesosomes participate in photosynthesis because they carry the photosynthetic pigment. In the case of nitrogen fixation bacteria, they have required enzymes in their mesosomes to fix nitrogen. In this way, the plasma membrane participates in various functions of the prokaryotes. Then, coming to the nucleus. There is a single large chromosome in chromosomes in all bacteria. So in all bacteria there is a single large circular chromosome. But a nuclear membrane is absent. Already I have told you the chromosome is naked without any RNA or proteins. The place of the cytoplasm in which the chromosome located is known as the nucleoid. So the chromosome is present in a place called nucleoid. This chromosome is also known as genomic DNA. Genomic DNA. Then, in addition to the genomic DNA, there are small circular DNA molecules. These are known as plasmids. In the genetic engineering, I have explained the role of plasmids in recombinant DNA technology. These plasmids are one to many. They are circular. They are made of double helical DNA, but they are independent. They multiply independent of the main chromosome. The multiplication of the plasmids is not based upon the main chromosome. They multiply independently on their own. The plasmids are helping us in biotechnology. The plasmids have antibiotic resistant genes. So, whenever they come across an antibiotic, they are not killed because these antibiotic resistant genes provide protection to the bacteria. In this way, they protect from fungi and bacteriophages. In the cytoplasm, we find 70S ribosomes. These are. I have told you 70S ribosomes are made of two subunits of 50S and 30S type. Then, in cytoplasm, we also come across reserve food materials. These reserve food materials are also not surrounded by any membrane. These reserve food materials contain phosphate, glycogen, and synophycean granules. These are all form the reserve food of the bacteria. Then, some bacteria also have flagella. But the flagellum of bacteria and eukaryotes differs. In the eukaryotes, the flagellum is made of 9+2 microtubules. But here, the microtubules are absent. The flagella may be one to many. The flagella has a basal granule, this one. Then it has a long flagellum and also has a hook. So the flagella of bacteria have three parts: a basal granule from which it arises, a hook, and a flagellum. But structurally, the flagella are different from those of eukaryotes. I will be giving a detailed structure of flagellum of eukaryotes later on. Then, the flagellum helps in swimming. Then, on the surface of bacteria, we find a number of small bristle-like structures. These are two types: pili and fimbriae. Pili are longer than fimbriae. They are structurally similar, but they differ in their length. Both of them help in the attachment of bacteria to each other or to the attachment to the substratum. That is the function of pili and fimbriae. In this way, a prokaryotic cell has various parts. So that is about the topic cell theory and prokaryotic cell. Thank you.